This is a lecture on DNA replication where we will discuss the origin of replication, replication forks, as well as several things that allow DNA replication to happen more quickly in our cells. And so DNA replication is the synthesis or the making of a new strand of DNA. And DNA replication has to happen relatively quickly. It actually occurs at a rate of about 3,000 bases or 3,000 nucleotides every single minute in humans. And even though this rate is quite high and nucleotides can be synthesized at this high rate, there's only about one mistake per 1 billion nucleotides that's made during DNA replication. And so that means that replication has quite a high level of fidelity. There's very little errors in this process, even though it's so fast. And so I'm going to talk a little bit about how replication can happen so quickly and several things that make it happen so fast in this portion of the lecture. And then in our next portion of the lecture, I will discuss how there's such a low rate of errors in the replication process. So replication can happen quickly because of these kind of three things listed on the slide. Replication is what we call semi-conservative, which I'll explain in a minute. It starts at multiple origins or multiple start points, and it happens in two directions, or it proceeds bidirectionally. And I'm going to explain more about each one of these three things on the next few slides. Right? And so the process of making a new strand of DNA or synthesizing DNA through replication is referred to as being a semi-conservative process. And so in a semi-conservative process, some of the original DNA is reused in the new DNA molecules. And so each strand of existing DNA, which you can see up here in this gray double-stranded DNA molecule, ends up serving as a template for a new strand of DNA, which you can see in blue. And so an old strand of DNA is split up into two individual strands. You can see those two gray strands here. And those strands are reused in new DNA molecules. And they're used to create these new strands in blue by acting as a template. And so by reusing one of the strands of DNA, you can actually create two DNA molecules, right, in about half the time, rather than having to synthesize two entirely new strands of DNA every single time you want to perform DNA replication. And so the idea of being semi-conservative means that one strand of DNA is conserved in those new strands every time that DNA synthesis or replication occurs. One other thing that makes DNA replication happen more quickly is the fact that replication starts at multiple origins or multiple start points. And so there is a region on your chromosomes where replication begins. And these regions are referred to as origins of replication. Origin meaning the start point. And the origins of replication on a chromosome are specific DNA sequences. They vary from species to species, but most of them tend to contain more adenines and thymines, or A's and T's, than cytosines and guanines. And the reason for that is that A's and T's only have two hydrogen bonds holding them together in the DNA double helix, whereas C's and G's have three hydrogen bonds holding them together. And so it's easier to open up a sequence that has more A's and T's than a sequence that has more C's and G's. And we're going to want to open up these origins of replication in order to have access to the DNA, like you can see down here in this picture, so that that DNA can be used as a template for synthesizing a new strand. So it makes sense that you want a little bit looser of a sequence, one that's easier to open up inside of your origins of replication. And what you can see here is there's actually been 4 origins of replication, each one of them occurring at a different place along the chromosome. And the fact that replication doesn't start on one side of the DNA molecule and then just proceed all the way down but rather starts at multiple points is one way that it can happen faster. Multiple start points is sort of like multitasking for DNA replication. What you'll also notice is that here's your origin of replication, and you can see two arrows in red inside of what's called the replication bubble, where replication is happening. And those two arrows indicate the direction of replication. And you can see that they're pointing in opposite directions from each other. And that's because replication also proceeds in two directions from the origin, or bidirectionally. And if we look at this image down here, we can see that origin of replication, and we can see replication happening in both to the left of the origin and to the right of the origin. And these structures here, these Y-shaped structures, where replication is occurring as the DNA unwinds, are referred to as replication forks. And these replication forks move away from each other in opposite directions and away from the origin, and then ultimately are able to continue replication until they meet and you've generated two pieces of DNA. And so the fact that replication is semi-conservative and reuses one old piece in the synthesis of a new piece of DNA, the fact that replication starts from many origins, and the fact that replication proceeds in a bidirectional way, or in two directions from those origins, all make replication happen a lot more quickly inside of our cells.